we will discuss about the MHC typing. So in case of humans, it is called actually typing. So in case of animals, we call it MHC. Actually, for humans specifically, it is called HLA typing. Okay. So my voice is clear to everybody.
Yes, sir.
Yes, sir.
Okay, fine. So just go to the presentation slide. Okay. So typing of MHC. So first of all, we'll discuss about what is meant by the major histocompatibility complex. So this particular protein, okay, it is a kind of protein that is present in every cell, in every tissues, and this particular protein is responsible for acceptance or rejection of the tissues when they are grafted. You just uh, take tissue or organ from one individual to another individual of same species. So what happens? There is every chance that another individual to whom you are going to transplant the tissue or the organ, it may reject. Okay, the body will reject. The rejection occurs because of the major histocompatibility complex protein. So in human beings, okay, the MHC genes, they comprise about 3.6 this MB. MB stands for megabases. 3.6 megabases of the DNA. That is 0.1% of the genome. That is the total genome of the human total human genome. 0.1% of the total human genome is comprised of the genes. Okay, that encode for MHC complex, major histocompatibility protein complex. So actually, MHC is not a single protein; it is a complex of many proteins. So that's why many genes are there. Okay, that are responsible for production of the MHC, and that is responsible for acceptance or rejection of the tissue. That's why the name is histo stands for cell or tissue compatibility. Okay, whether so this protein is responsible for acceptance of new tissue or cell in a human or animal body. So this in case of human beings, this particular MHC genes, they are located in the short arm of the chromosome 6. The 6th chromosome is located. Okay, and MHC is most gene dense region of the human genome. Okay, now what is meant by gene dense region? So let me open a whiteboard. So supposing that say this is the DNA, okay, or complete genome. Although the genome, this animal genome or mammalian genome, they are divided into chromosomes, but I'm just putting it in the same one molecule. Let us assume that this is a complete genome. Okay, and in the genome, okay, you will find that some specific region of the genome, okay, okay, so you will find the genes. Say in this case, the green portion, okay, it contains the genes, okay, and the rest of the black portion of this DNA, okay, these are not genes, okay. So now the thing is that this kind of gene rich, I would say that this particular region is the gene rich region of the DNA. Okay, it is gene rich region of the DNA. So now this kind of gene rich region of the DNA, it spans many part of the genome. It is not that this kind of region is located in only one part. Okay, so it is located in different parts. Say for example, if it is this is one gene rich region, okay, and similar gene rich region you will find somewhere else also. Okay, so now as per the definition says that this MHC genes where they are present. So that region is rich in the genes. Okay, that means the genes are quite close together. Okay, okay. So now we discuss about the structure and function of the MHC molecules. So here I have written HLA. So HLA actually it is for humans. So normally it is called MHC. The MHC molecules present in the MHC molecules present microbial and viral peptides to the T lymphocytes, which then mount specific immune response against the invading pathogens. That means if I just go to this, so normally what happens? This is a cell, okay, and when any kind of pathogen, say this is a pathogen, so I would say it is a pathogen, okay. So now this pathogen, when it enters the cell, okay, then this pathogen it enters the cell in a particular fluid filled cavity that is called endosome. So it will be inside an endosome, okay, and then that endosome now it will fuse with the lysosome. This is a lysosome. It will fuse with the lysosome. So here what will happen? This particular this antigen or microbe or virus, whatever it is. So what will happen? This will break into multiple pieces. So we call it antigenic peptides. Okay. So they will break in the multiple pieces. So these pieces of this particular virus or protein, antigen, whatever it is, so these are called antigenic peptides. Now these antigenic peptides, now they are loaded into a MHC complex. So now these are they are loaded into MHC complex. So here we load it here. Okay. So this MHC complex it has got a particular cavity or it got a particular pocket where these antigenic peptides will be loaded, and after that this will move to the cell surface. Okay, and then this will be recognized by T cell. Okay, this is a T cell. Okay, T cells it has got a particular receptor. So this particular receptor it is called T cell receptor. Okay, so now what happens? This T cell receptor this will bind the MHC complex bound with the antigenic peptide. So one thing is to remember that this particular T cell receptor it will not bind empty MHC. Okay, it will bind only the binary complex. Binary complex means the complex made of MHC as well as the antigenic peptide. Okay, so now this may be a cytotoxic T cell or it may be a T helper cell. So that I'm not going to discuss right now here because this is beyond our scope of the scope of this course. Okay, so now this particular T cell now it will show the activity once its T cell receptor binds the binary complex. So this is what is the major histocompatibility complex. Now the major histocompatibility complex it is of two types. There's got two classes. So one is what we call class 1. Then we have the class 2. And there is another class that is called class 3. So class 3 is mainly present in the NK cells, that is the natural killer cells. Okay. So the class 1, it is present in all nucleated cells. Okay. Then class 2, it is present only in antigen presenting cells. That may be macrophages, B cells, because B cells also sometimes they act as phagocytes. Okay, macrophages, B cells, and all. Okay, or broadly we call them APC, antigen presenting cells. So the class 2 is present only in the antigen presenting cells. Okay, but the this class 1, it is present in all the cells. Class 3 is present only in the natural killer cells. So I'm going to discuss about this class 3. So class 4 and class 2 will discuss. Okay. So now this is the just in the continuation of the this structure of the MHC molecules. So you can see this is the DNA. Okay, that is, and these are the genes: alpha 1, alpha 2, alpha 3, TM, C, and this is C. So they have 1, 2, 3, 4, 5, 6, and 7. So not 7, 6. Okay, so we have 6 genes. Okay, these 6 genes that lead to production of the this MHC complex. So what happens? See, these are the genes and they are away from each other. So this part of the DNA, you can see, this part is the non-genic DNA. Non-genic means this part of the DNA is not genes. Okay, so these are the first gene, second gene. Now what happens after transcription? Okay, you know that in case of eukaryotes, after transcription, another process occurs that is called splicing. So that means you can say that this part of the DNA, okay, this one, this one, this one, okay, this part of the DNA, so this is actually introns. Okay, then they are removed and then it is produced as one mRNA molecule. Okay, from this one mRNA molecule, okay, this will produce complete protein. Okay, so now the alpha 1, okay, is responsible for making the this particular domain. Alpha 1 domain, alpha 2 is for alpha 2 this particular domain. Okay, alpha 3, TM is the responsible for transmembrane. Transmembrane means this part of the protein is present inside the membrane. Okay, and then we have this C stands for cytoplasmic domain. That means this part of the protein is present inside the cytoplasm because this is the cell. Okay, this is the cell. Okay, and here at this point, okay, you have the cytoplasmic domain. Okay, and this part is a part where it binds the antigenic peptide. Okay, and then the second thing about this one is the this particular structure of the protein. Okay, it comes from beta 2 microglobulin. So this particular protein is different. It is not coming from this group of genes. It is a separate gene. Okay, that after synthesis the protein gets attached here. That means what I'm saying is that we have two types of proteins. Okay, one is monomeric protein. Monomeric means having only one polypeptide. Then we have multimeric proteins. So multimeric proteins means proteins having more than one polypeptide. If you have only two polypeptide, we call it dimeric proteins. Okay, again dimeric proteins or multimeric proteins, there are two types. One is called homodimers or homo multimer, and one is called heteromultimer or heterodimer. Okay, you can see here in this case the MHC. Okay, this particular protein. Okay, it is a heterodimer. So why it is heterodimer? If we just see the structure from here to completely this one, it is a single protein. Okay, then this one, it is a different protein. Okay, so you have one polypeptide here, and this is the second polypeptide. So it is a dimer. So heterodimer means both the dimeric units are different. Okay. Similarly, you have the MHC class 2 molecules. Okay, how the MHC class 2 molecule is produced in the same way. Okay, they are away from each other. Okay, and then they make the complete structure. Okay, now in this case, what you find is that in earlier case, this one domain was very big. Okay, and the beta 2 microglobulin domain was smaller domain. Now in this case, okay, you find that the beta 2 domain, it is as big as the alpha domain. Okay, and here this antigenic peptide binding domain, it is made exclusively by the alpha domain. Okay, beta domain is not contributing in the formation of this particular pocket. Now in this case, what happens, both alpha and beta, they contribute in formation of the pocket. This is alpha domain, this is alpha 1 chain. Okay, this is the beta domain. Okay, this is the beta chain. Okay, fine. Now this part is responsible for making the pocket for holding the antigenic peptide. So this is just another diagrammatic view. So what you can see here, this is a class 1 molecule. So this is a alpha 1 domain, complete alpha 1 domain, and is alpha 2 and alpha 3 domain. Okay, in this domain you have the peptide binding cleft. So this area, it binds the antigenic peptide. So beta 2 microglobulin is a small domain. Okay. Now, if we look into the MHC class 2, this is class 2 molecule. Okay. Now, in this case, you have alpha and beta both of equal length. Okay. And then this alpha and beta, these two are separate proteins, and then they combine to form the peptide binding cleft. Okay. But in this case, this peptide binding cleft is made by the same protein. Okay. And then next thing is that what you observe here is that this particular this structure is bound only by alpha domain. Beta domain is simply attached to the this particular alpha domain. It is not having any kind of direct link with the membrane. But in this case, the beta domain also has one transmembrane domain. Okay. Now, the major difference between the class 4 and class 2 is that the ends of peptide binding groove are more open in class 2 molecule than in class 1. That means this is class 2 molecule. Say, if you see here, this particular peptide binding cleft, it is made by two different proteins. So that is why their the size is bigger. And in this case, it is made by same protein. So that's why the cleft size is smaller. As a result, what happens? The MHC class 1, it can bind only short peptides. The peptides having only 9 amino acids. That means the peptides having more than 9 or 10 amino acids, so they will not enter into the MHC class 1. They cannot hold that kind of bigger peptides. And MHC class 2, they have got the capability, okay, to bind the longer peptides. That is of 12 to 20 amino acids they can bind. Okay, now we talk about the MHC polymorphism. So first of all, briefly, let us discuss what is meant by polymorphism. So polymorphism means a difference in a structure of a gene or difference in a sequence of a gene in a population. So now, but nowadays the word gene is not used because there are a lot of non-genic DNA, okay, which are not genes and they are polymorphic. So better to say that it is the variation of a particular DNA sequence in a population. So for example, in a particular locus, we have say blood group AG in one individual. Another individual in the same locus, it has got blood group B gene. Third individual, the same locus, it has got gene that is non-functional. So you call it a O blood group. Okay. So that's what is the polymorphism. So that means in the same locus, okay, in a population, okay, the gene sequence or the DNA sequence it varies. Okay. But I'm using many times gene sequence. Actually, it is not always the genes. It may not be genes. There are many non-genic DNA also. Their sequences also vary in a population. So they're also polymorphic. Okay. So here what it says is that MHC genes are highly polymorphic. That means the in a population, okay, you will not find any individual, okay, having similar MHC sequence. Even the MHC sequence of a child is different from his father or mother. Okay. Now it says this statement says the N terminal part is most polymorphic. So if we just look back, just look at this structure. Okay, this is the MHC class 1. This is the C terminal domain that is present inside the cytoplasm, and then it goes like this, and this is the N terminal domain. Okay, so it says that this N terminal part is more polymorphic. That means the sequence of the amino acids in this N terminal, it is highly variable in a population. Okay, but other parts, say alpha 3 domain, they may be similar in different individuals in a population, but this part is highly variable. Okay, then the most polymorphic regions, okay, in class 1 and class 2, okay, it tells about the exon 2 and exon 3 in class 1 and exon 2 in case of class 2. Okay, because we have discussed earlier that in case of eukaryotes, you have a gene. I mean, in case of prokaryotes, you have a gene. Then the length of the final RNA, mRNA, is equal to the length of the gene. Okay. But in case of eukaryotes, in mammals, what happens? You have this much big gene. Its final mRNA will be shorter because you have a big gene. Then you have a big mRNA. It undergoes splicing, removal of the introns, and then it becomes shorter. Okay. So then here what happens? The introns are removed. So you have exon 1, exon 2, exon 3, exon 4 like that. Okay. So exon 1, exon 2, exon 3, they just combine to form a mature mRNA. Okay. So here it talks about the exon. Exon 1 means first exon. Okay. So exons are numbered as first exon as exon 1, second as exon 2. So exon 2 and exon 3 of the class 1 and exon 2 only for class 2. So these regions are highly polymorphic. That means if you sequence this region, okay, you'll find that every individual they have different sequence of the DNA at that particular region. So this particular region of the MHC genes, class 1 or class 2 genes, they are focus of the DNA based MHC typing. So now, okay. So now there are these three are the typing methods. Okay. The first is the sequence specific PCR. Okay. So in this case, in the sequence specific PCR, what is done? Say this is the class 1 or class 2 MHC gene. Okay, and then say this part is, let us assume that this part is the exon 2. Okay, which is highly polymorphic. Okay, now you want to do the PCR. Okay, so what it says, sequence specific PCR. So now what you will do, you will take the primer. Okay, I assume that you have good knowledge of PCR. So one primer will get attached here. That's what you call, say, forward primer. Then another primer you have, that is called reverse primer. Okay, fine. Now you are developing these primers. Okay, because you should have the prior knowledge of this complete sequence of the DNA. Okay, so now based on that, you made the primer. Okay, you made the primer and then you did the PCR. Okay, so this primer will get attached here. So this will get attached here, and likewise, sorry. So likewise, you will start getting the amplified product. Okay, so you start getting the amplified product. Okay, now here there is an issue because this particular primer will this primer set will carry out PCR. Okay, it will carry out amplification only when these two primers are specific. The thing is that the exon that we are trying to amplify using PCR, okay, this particular exon is highly polymorphic. Okay, so what will happen? Okay, in some individuals, okay, these primers, they find it difficult to get attached because the attachment of the primer to the ends of the gene, it is through the base complementarity. That means if you have A here, you should have T here, like this. Okay, but as the whole of the DNA, it is highly polymorphic. As whole of the DNA is highly polymorphic, so as a result, what happens? It is possible that in some individuals, okay, this part and this part of the DNA where the primer binds, okay, it it may be highly different, or it may have the variation. So in that case, if this part of the DNA where the primer binds, it is different from what you expected. So in that case, what will happen? Your primer will not bind properly. So in that case, amplification will be less. Okay. Now in this case, what will happen? How you can make the judgment? Okay. So just you take a primer set. This primer set you have developed, okay, based on the knowledge of that particular sequence. Okay, you've taken one sequence, okay, and then based on that you made the primers. Okay, so in that sequence, when you will do the PCR, your primer will bind properly, and you will get a lot of amplification. You take the DNA of some other individual. Now that individual will be having some difference in the sequence. So now the primer where it binds, that part is also bit different. So what will happen? Your primer will not bind properly in the second individual. So what will happen? The amount of amplification you are getting will be less. Okay. So now more is the difference. Okay. Lesser will be the product. Okay. So you have the more difference and the lesser will be the product. Okay. So that is what is the sequence specific PCR. So in this case, what you will do, you will develop a primer. okay, based on a particular sequence of one individual, then you will use that primer for every individual. Okay, so now any individual which sequence is similar to this individual will be having more amplification. All and other individual whose sequences are more different from this individual, they'll be having less amplification. Okay, so now it is based on how much amplicon or how much amplified DNA you are getting. Okay, so based on that we do the typing here. Then second one is the SSOP, sequence specific oligonucleotide probe. So in this case, what happens? So here, what is done? We don't use the primer specific for the end of this particular DNA. So this red colored DNA is the highly polymorphic DNA. What we do, we make a set of primers, okay, that is specific for this end of the DNA. Okay, so now what happens? We are using this end of the DNA. So this end, because this part of the DNA, it is highly polymorphic. That is responsible for, there is one of the exon, highly polymorphic exon responsible for MHC. Okay, but this part of the DNA, this is not polymorphic. It is same in all the individuals. Okay, but this part is highly polymorphic. It is different in different individuals. So you make a primer against this part of the DNA. Then when you will do the PCR, so what will happen? The amplification will be like this. Complete amplification. Okay. So okay. Now after making the amplification, now in this case, what will happen? You take any individual, every individual will be having a lot of amplification because you are not using the primer against the region of the gene which is polymorphic. You are using the primer against that region of the DNA which is less polymorphic. Okay, fine. So in that case, primer will bind properly in all the individuals in a population, and then you get the amplified product. Okay, now you use the probe. In this case, we use the oligonucleotide probe. That means what we do. Let me open another thing. Okay. So these are the amplified product. You run this amplified product in a agarose gel. Okay, and then what you will do to this DNA. Okay, now you will use the probe. So probe is just like primer. Okay, so it is a single stranded DNA. Okay, so which is single stranded DNA that is specific for this region of the DNA. Okay, so what is done? Say this is the probe which you developed. Okay, and then what you do to this probe, you attach a fluorescent dye. Okay, and then put this probe here. Okay, the thing is that if this particular probe you have developed, if its sequence is exactly complementary to this particular sequence, so this probe will this particular fluorescent dye will fluoresce. Okay. Now the thing is that if the complementarity of this DNA and this DNA, if it is less, what will happen? The intensity of the fluorescence will be less. Okay. That means, say you have made the amplification, you have a lot of product, run it in the gel. In the gel, after that, you just put it in perform the southern blotting. Okay. Oh, sorry. Okay. So just take the do the PCR, perform amplification. Okay. After that, what do you do? Just run the DNA in a agarose gel. Okay. Then blot this DNA into the, say, nitrocellulose membrane. Now in the membrane, you have the PCR product or DNA. Now in that DNA, you add the probe. Now this probe is complementary to the polymorphic DNA. Earlier, what we have seen is that the primer which you took for PCR, this was specific for the non-polymorphic part. Okay. After PCR, after southern blotting, okay, now you have the DNA, amplified DNA PCR product in the nitrocellulose membrane. Now in that membrane, now you add the probe. Now this probe is just like primer. This will get attached. Now this probe is specific for, or you made this probe specific for polymorphic DNA. So in the polymorphic DNA, this probe will get attached. Okay. The thing is that now here the point comes. If your probe is 100% complementary to the polymorphic DNA, then it will give highest fluorescence. If the probe is less complementary, that means you have A here, in your DNA you have T. Okay. Then next you have T in the probe, and here instead of A you have G. That means there is no base pairing. But again, third one is base pairing. So somewhere it is not properly base pairing. That is lack of complementarity. So in that case, the fluorescence will be less. That means the probe will not bind the DNA properly. Okay. So that is the basis. Okay. How you can find out the difference of you can find out the polymorphism of MHC in a population. Okay. Now we go to the third one. The third method is the sequencing based method. So sequencing based method is quite straightforward. So in this case, what is done? So same way, here we take the primer specific for the non-polymorphic part of the DNA, okay, and then we get the amplification. After this, this DNA, it is sent for sequencing. So this DNA is sent for sequencing. sequencing. So then you find out the sequence of this. You find out the sequence of the polymorphic DNA. Okay. So the third method appears to be more appropriate, but it is a bit expensive. Expensive because sequencing it is a costly affair. The first two methods do not need sequence. There is no requirement of sequence. Okay. So the first method, what you are doing is that you are using a primer specific for the polymorphic DNA. Okay. So your primer will get attached properly only when the primers are exact complementary to the region where it binds. Okay. So in that case, you will get large amount of amplification. Now you have a DNA where the sequence is bit different. Your primers will not bind properly. So in that case, there will be less amplification. So how much amplification you are getting? So based on that, you can type. This is type 1, this much amplification. This is another DNA, this much amplification. This is another DNA having this much amplification. Then in the second method, what you are doing is that you are using the primer specific for the non-polymorphic part of the DNA. So this is a polymorphic part and here non-polymorphic part. And then always when you do the PCR, always will give large amount of amplification. Okay. Now what you will do after PCR, okay, you will use the probe. This probe is just like primer only. This probe you will add it to the DNA. Okay, and this probe contains a fluorescent dye. If your probe because this probe is specific for the polymorphic part, so this probe will bind the DNA. If the binding efficiency is very high, then you'll get very high fluorescence. In what case the fluorescence will be high, or in what case the binding efficiency will be high? When the sequence is exact complementary to the sequence of the probe. Okay, and the third method is straightforward. Just you do the amplification and send it for sequencing, and you know the sequence and find out that okay. So now, what is the application of MHC typing? So MHC typing is required for organ transplantation. Say you are going to transplant an organ from one person to another person. So how will you know whether that person will accept it or not? Okay, or at what level of acceptance? Generally, it is said that 80% similarity. MHC, if the 80% of the MHC is similar, then it's okay. It will the organ will not be rejected. Okay, if it is less than 80%, then organ will be rejected. Okay, so this MHC typing is mainly used for your organ transplantation.